Hello all, I will just announce this in the group. Then we will start with today's translation lecture. Okay, so today we have about to start our next point in the syllabus, that is the translation. So we'll wait to 1 or 2 minutes more and then we'll start. Okay. So we will meet 1 more minute. So let's see. Well, others let join. So it's 11:30. We are having today's not very much theoretical lectures. So whatever these lectures are, I'm going to conduct. Most of them will be like a shorter versions, and you can actually understand this translation by because what I'm going to include is the detailed diagram on this. Okay, so that it will be easy for you to not only just remember this translation but have a detailed idea about it. Okay, so let's start with today's first point, that is this translation. See, in this translation, to initiate this translation means what translation is. We have already seen the DNA replication and DNA transcription. In this DNA replication, what was the case is that we have replicated our DNA. Okay, we have formed two copies of our DNA in this DNA replication. In the next process, that is the transcription, what we have seen is we have just made an mRNA or the RNA from this DNA. That was called as transcription. Now what we are going to see? Now we are going to see translation. We will translate this RNA into protein. So this is called as what? Translation. So that is the thing. Let's see. Yes, good morning, Zishan. Good morning, others also. So let's start. So in this, to translate or to start this protein translation or to start the translation of this RNA into protein, we require some initiation codons, okay, and some termination codons to stop this entire protein synthesis. So let's start these. What are these initiation codons are? See, initiation codons are mostly this AUG, or sometimes these initiation codons can be GUG also. And where these codons will be present? These codons will be present on this, right, RNA. Okay. So let's see the first, and these are the initiation codons. Then there are also termination codons are also there. Okay. And what are these termination codons? These termination codons are just stopping this entire synthesis of protein. Okay. Or entire just translation of protein. So that's why they are called as UA. These are called as nonsense codons because they don't code for any protein. So that's why they are stopping this entire protein synthesis. So that's why called as they are also what? UAG, UGA, or UAA. You will have to remember all of these four examples. This, and this initiation codon. Okay. Then this codon anticodon reaction, what is the thing is that, see, this is the mRNA, right? On this, the codons are present, and there will be another RNA that is called as tRNA. Okay, it will have a structure like this, and it will have an anticodon arm to it. In a structure, tRNA is having anticodon arm. Now, this what is there are almost seven nucleotides will be there. These seven nucleotides will function combinedly and recognize the three codons on the mRNA. And finally, with the help of these seven nucleotides, amongst those seven, only three anticodons will make a bonding with this codon. So that is what this codon and anticodon recognition process is. Then there is one hypothesis is there that is called as wobble hypothesis. Now let's see what is wobble hypothesis is. Okay, see, wobble hypothesis just nothing but means just a single tRNA. Okay, one tRNA molecule can recognize more than one codon. That means what? You suppose this is that tRNA. Okay, Usually what should happen, as this anticodon has three nucleotides, this codon has three nucleotides, so there must be only a one. Suppose this is a u g. Okay, so the combination must be what? This suppose uracil, adenine, and cytosine. This must be the combination on this codon, or this must be the combination of anticodon. Okay, this can be a possible codon anticodon possible, only one. So usually, what is thought is that there is one single RNA or a single tRNA should recognize only one codon, but that is not the case. That is not the case. Why it is not the case? Because this third nucleotide, okay, because of there is some incapability of this third DNA, third nucleotide base, this one tRNA molecule can recognize more than one codons. It can also recognize, say, for example, XY. Suppose the anticodons XYZ. So they can recognize A, B, C also. They can recognize P, Q, P, B, C also, or anything else. So the case is that they are recognizing more than one codons. So that is what the Wobble has given, that this tRNA can recognize more than one codon. Okay. So that's why there are less number of tRNA are present as compared to the even these other codons which are for proteins are there. Like suppose there are 60 codons for the proteins, triplet codons for the proteins, but there are almost around only 40 tRNAs are there. This is why, because this one tRNA can now identify and code more than one codon. Okay. So that is what this Wobble hypothesis is. Now let's see what is the degeneracy of the genetic code. See, the degeneracy of the genetic code means what? The existence of multiple codons for a single amino acid. Means what? Suppose this is the A, B, C codon is there. It can also code for this X amino acid, and this is the another you can say PQR. This can also code for the same amino acid. So in this way, there is existence of multiple codons for a single amino acid. This is called as what? Degeneracy of the genetic code. Okay, more than one codons can code for a single amino acid. Okay, so this is the basic, you can say, meaning of this Wobble hypothesis and degeneration of the genetic code. Then we are going to actually start this entire translation, but before that, there are certain things which are required to start the actual translation. No, this is the case with the prokaryotes. Okay, this is the translation in case of the, sorry. First, we are going to see the translation in case of the eukaryotes. So let's see what are the requirements. See, obviously, as the protein is going to be formed out of the amino acids will be the first requirement. Then the manufacturing machinery, that is ribosomes, actually form a functional complex and synthesize the entire protein. So obviously, they are one of the most second most requirement for this protein synthesis. Then these are the different types of ribosomes are there. 70S are there in these prokaryotes, 80S are there in the eukaryotes. Okay, so this will depend on the which translations we are going to see. We are going to see first the eukaryote translation. Okay, so in that these 80S ribosomal units will be present. So there are different sites. There would be different sites on this ribosomal unit. Okay. Usually, initially it was thought that this acceptor site A and this peptidyl site P site, or the binding of peptidyl tRNA site P site. These two sites are only usually present in the eukaryotes. But right now, nowadays, these E site exit sites are also shown to be the part of even the eukaryote ribosomes also and prokaryote ribosomes also. Now we can show all of these three sites in all the three types, all the two types of translation, whether that is prokaryotes or eukaryotes. Okay. So what are the different sites on these ribosomal units? In this, what will happen? This entire will be the what? ATS ribosomal unit. Okay. So let's see now. This will be the A site is also called as an acceptor site because it is accepting this amino acid tRNA. Okay, this is the P site. You can also call it is because of this peptidyl tRNA attachment, or you can it is because of this it is forming an peptide bond with the next A site tRNA. The next site is E site is quite easy. It is just an exit site. Okay. So let's see now what are the other requirements. Obviously, the mRNA after which is, we can say, a transcript of this entire translation. You are reading this mRNA codons, and then you are only synthesizing the protein on depending on that mRNA. Then they say there this is tRNA. These tRNA are just carrying all these amino acids which are required. Okay. See, as this entire protein is going to be formed out of this amino acid, one amino acid, suppose two, then amino acid three. So such long chain of amino acid is going to be formed out of what? Out of these amino acids, and then you are calling it as a protein. So these amino acids, these individual amino acids are carried over to this machinery by this tRNA. Okay, so obviously they are going to require. Then there must be the requirement of energy. So the energy can be obtained in the form of like this adenosine triphosphate (ATP) and guanosine triphosphate or the GTP. There are certain other protein factors are there, you can call it as enzyme coenzymes. Okay, they will be, obviously they will be required for the initiation of this translation, elongation of this protein peptide chain, and obviously for the termination, means to stop the protein synthesis after the desired length of protein is uh, achieved. Okay, now we are going to see, we are going to see just the diagram part of this eukaryotes and prokaryote translation, because if we go much in the theory, that is going to be quite a bit, you can say, boring also. So I will just explain you the entire process in this diagram. Okay, so that you will understand. See what will happen in this way. This is the diagram for the initiation phase, right? We are just initiating to synthesize the entire protein. So what are the things that are happening in this initiation? See, this is the ATS ribosomal units in the eukaryotes. We have already seen. But if consider their subunits, they are sorry, formed out of one larger unit and one smaller unit. Okay. So the larger unit is 60S ribosomal unit, and the smaller unit is what? 40S ribosomal unit. Now, what will happen in the initiation phase of this translation is that this ATS ribosomal unit will dissociate in two forms. Okay, and why it is dissociating? Because see, this whatever this functional ATS ribosomal unit complex is there, that cannot be formed directly from this ATS ribosomal unit. Okay, means what? This ATS ribosomal units will need an attachment of some protein machineries to it. Okay, some proteins, enzymes, GTP, ATP, all they will be required to attach to this ATS, and then it is you are forming this functional ATS ribosomal unit. Okay, but this formation of functional ribosomal ATS ribosomal unit is not going to occur after the, if these ATS are present as such in time. Okay, they should be first be get divided into their individual units like this 40S and 60S. Then what will happen? These 40S subunits will carry most of the working proteins along with it, and after that 40S ribosomal unit along with the not only working machines, not only working proteins, but it will also carry this mRNA with this 40S only, and that complete unit then will fuse with this 60S. Then you will finding you will get this functional or the ATS initiation complex, that is can actually start the protein synthesis. So let's see now how can this 40S ribosomal unit is carrying, or it is just occupying all the proteins and even the mRNA onto it. Okay, so the first step, that is this there are some initiation factors are there. Okay, initiation factors means what? They initiate this entire protein synthesis. They are called as this initiation 3 or 1A. Okay, so these initiation factors, okay, these initiation factors 3 and 1A will first attach with this 40S ribosomal unit. And what's the additional significance of this initiation factor is that they also did not allow this 40S ribosomal unit to immediately form an reassociation with the 60S ribosomal unit. Okay. So that is also prevented with the help of these initiation factors. The next thing that is going to occur is that this initiation factor, now this 40S pre-initiation complex along with this initiation factor 3 and initiation factor 1A now get combined with this methionine tRNA. Now what is this methionine tRNA? This is the tRNA molecule which will have a methionine molecule attached to it, methionine amino acid attached to it. Along with that, there are some other initiation factors like this initiation factor 2 is there, and one GTP molecule is there. So this is one of the, you can complete complex which will have not only the tRNA, but it will also have a source of energy for the entire reaction. So now this tRNA will attach with this 40S ribosomal unit. Then this will form this initiation complex, 40S initiation complex. It is having what? It is having this 40S ribosomal unit, this initiation factor 3, initiation factor 1A, then this methionine tRNA, then this GTP, and this initiation factor 2. Now all of this complex is now going to attach with this mRNA. Okay. This mRNA is also having some required some additional factors to act upon it. So these factors you can actually exclude these factors if you just cannot digest it. But I will just tell you in the short. See, the first is that at the cap, means at the 5' end of this mRNA, with the help of this energy from this ATP, there are some other factors are there, like this 4F factor is there. It's going to attach at the 5' end or the cap of this mRNA. So that's why then it's going to be form this mRNA, which is now ready to be fused with this 40S ribosomal. Okay. So this mRNA along with this 4F initiation factor now attach with this ribosomal units, or now form a complex with this ribosomal unit. Along with this, you are utilizing the energy in the form of ATP. So now you have this. Now this is the entire what you can call as 40S initiation complex. Now this initiation complex is now going to directly fused with this whatever this earlier 60S ribosomal units was there. Now it is going to fuse with this complex. Okay. Because now this is this 40S is carrying all the other initiation factors. It is also carrying this tRNA. It is also carrying mRNA. So now, as to allow this combination of this 40S ribosomal functional ribosomal unit and the 60S ribosomal unit, the earlier this initiation factor 3 and 1A was preventing the association with the 60S. So now it will get removed. Once it get removed, the initiation factor 5 will come into play, and now it will just fuse both this 40S and 60S, and it will form this final 80S initiation complex. Okay. As this 80S initiation complex is formed, we are ready to go for the protein translation. Now let's see what's happened in the last part of this translation, that is the elongation and the termination. Sir, I think there is some query. Sir, if possible, you provide your contact number. I have some queries regarding the strategy of zoology option. Okay. See, my contact number is already there in the public domain already, both of them. But still, if you want, you can have this. Okay, 9096468518. Okay, it's already there. Both numbers are there. So let's see what is happening in this elongation and the termination. So we have this initiation complex till now, right? This ATS initiation complex. But right now we are going to form the protein, and also we are going to elongate the protein chain. See, because the protein is going to be formed after this attachment of multiple amino acids. Okay. So the first thing would be the initiation to attach the first amino acid. And then the next phase would be to elongate this chain length, and the final step would be what? To terminate the once we have achieved the desired length of this amino acid or desired length of this protein, we are going to what? Terminate this protein so that we can have a protein of proper length. So now let's see how this elongation is occurring. See, we have seen this here also. You can draw E site also. Okay, no problem. Okay. So the next thing is going to happen with this ATS initiation complex is that with the help of energy from this GTP, this another tRNA, second tRNA which is carrying amino acid tRNA along with it, will attach with the help of this elongation. Now initiation factors were there earlier, now they are elongation factors. So the elongation factor 1 alpha and this amino acid tRNA, and with the use of energy from the GTP, this entirely will bind to what? Will entirely bind to this ATS initiation complex. Once they are attached, you can see now this is the methionine tRNA earlier which have bound. This is bound to this UAG, means initiation complex. Then the next tRNA is carrying this amino acid 1. It is attached to the where? A site. Now the next enzyme that is going to act is peptidyl transferase. Now what will this peptidyl transferase will do? This peptidyl transferase will form a bond with this methionine and amino acid. So that's why it is attach with this peptide bond formation. Okay, now this peptide has been formation. The peptide bond has formed. Now you can see this tRNA is not having anything. Okay, attached to it. So this tRNA can be removed from this entire initiation complex or the protein synthesis, and that removal of this tRNA not having anything will occur from which site? That is the site called as exit site, E site. Okay. So in this way, the things will happen. Now you can see this tRNA carrying this amino acid and methionine is now at this P site. The another tRNA will come, which will having another amino acid, and it will attach to this A site, and further this attachment of another amino acid will occur by forming this peptidyl transferase induced peptide bond, and in such a way, this elongation is going to occur. So after the multiple repetition of such steps, we are going to form this our ultimate protein, which will having what? Which will having this all of these amino acids attached to it. So now this, as we have elongated this chain, now we also have to terminate this protein chain, right? So that termination is going to occur with the help of this UAA termination codon. You can also have UAG or UGA also. So once these codons occur on this mRNA, this entire protein chain is going to be terminated. Okay, and after this termination, you are getting this protein separately. You are getting this 40S ribosomal unit and 60S ribosomal separately, which are ready for the next round of protein synthesis. You are also getting this mRNA. Okay, and this tRNA is also getting released. Okay. So this is how this entire translation in eukaryotes is. Okay, now we are moved to this translation in case of the prokaryotes. Let's see. The only one case with this prokaryote translation is that, see, in case of the eukaryote, the mRNA was monocystonic. Okay, means what, the mRNA can only synthesize one protein. But in case of this mRNAs of the prokaryotes, these are polycystonic. What this polycystonic means? These mRNA can synthesize, or these mRNA has many coding regions that can code for different proteins. Now let's see how the initiation is occurring in the case of the prokaryotes. See, the initiation in the prokaryotes is happening with the involvement of, obviously there are going to be formed different subunits as in the case of eukaryotes. So in case of this 50S ribosomal unit and this 50S and 30S ribosomal are going to be formed. Okay, out of that 70S ribosomal unit, yes. Okay, so for after this you can say dissociation of the ribosomal unit, this 30S will attach with these initiation factors IF3 and IF1. Okay, same as in the case of eukaryotes, these factors are also going to prevent this association of 30S with this 50S until this 30S is carrying most of the enzymatic complex or the protein complexes which are required for the translation, and also the mRNA also. So once these IF3 and IF1 are attached, you can see I have shown it here like this. Right? You can also use such trick in your diagrams. So now this attachment, this pre-initiation 30X complex will now get attached with what? With this mRNA. And where they will attach, they are called as promoter region. So in case of this prokaryotes, that region is having these codons like these sequences like the Child-Dalgarno sequence. So at that point they will get attached. At the meantime, this amino acid methionine tRNA is also getting activated for the attachment with this entire 30S ribosomal unit. So it will also get attached with this GTP, and there will be another initiation factor will be involved, which will attach itself to first methionine tRNA. It is called as initiation factor 2, right? So this initiation factor 2 along with this methionine tRNA and the GTP now will attach to what? To this 30S ribosomal unit, which is already attached with this mRNA. Then after this both combines, you will getting this complex, which is called as 30S initiation complex. Now what's going to happen? Now this whatever these initiation factors 3 and 1 were there, they will get released. After their release, they can now form an attachment with this 50S ribosomal unit. So that's why you are getting here your final initiation complex, that is called as 70S initiation complex. Now let's see what's happening in this elongation part. See, this same thing is going to be occur as in the case of eukaryotes only. This P site will have this methionine tRNA, and the initiation codons is like AUG. The A site is the site where this amino acid tRNA will get attached, right? Then after this attachment, in the next phase, you can see this peptidyl bond is formed, right? Peptide bond is formed. In the next phase, what will happen? This Okay, this tRNA will shift to this E site. This A site tRNA will shift to now this P site. Now A site is vacant. Okay, so what is going to happen? This needs to be get removed, and this there will be an addition of another tRNA at the A site. Okay, and remember one thing: both E sites and A sites cannot be occupied at the same time. Okay, so that's why when this E site gets vacant, this A site will get occupied with the next amino acid tRNA. So this is the case. Now, once this are returned, it is again going to form an attachment or a peptide bond with this newer amino acid. So, in this way, this chain elongation is going to occur. Now, you will have this final peptide chain at your tRNA of your P site. But how this termination is going to occur in this case? In this case, there are certain release factors are there, and which are also attached themselves at these termination codons like the UAG, UAA or UGA. So at these termination codons, these release factors will be present, and these release factors will finally cut down this entire peptide chain. And when they cut down this entire peptide chain, after that you will have this individual protein, this 30S ribosomal 50S and this tRNA separately. So this is how this entire translation is in the case of prokaryotes. Most of them are same, say, alike only, similar only, but you won't find this differentiation in any books. So I have just simplified for you. So you can just go through this diagram only and you can write most of the theory. Okay. So if you have any query, you can ask. I will just wait for 1 minute so that I will attend your queries. Do you have any queries or no? Okay, you have queries, you can contact me directly or you can just give your queries here also. I will attend in this live sessions also. Okay, and as I have already completed the cell biology module, we are about to start our next module around this 1st December. So I will release the date for this fees payment also. Today only I am going to just launch those the videos also, which you have in which we will have the idea of entire how the entire cell biology course went. Okay, so I don't think there are any queries. I don't see. So we will stop here and we will meet tomorrow at the same time 11:30 only for our next lecture. And tomorrow we will going to see I think about the chromatin. I will announce these prior these topics which we are going to cover tomorrow prior. Okay. So till then, have a great day. Bye bye.